I'm Shinsuke Niwa from Tohoku University. My interest is molecular motors, especially KIF1A, because since I was graduate student, I have studied KIF1A from knockout mice to C. elegans.
Studying KIF1A in C. elegans, which is a worm model, what are the advantages of using a system like that for KIF1A research?
The advantage of using C. elegans is we can make disease model C. elegans by introducing patient mutations into worm by genome editing. And also, we can do suppressor screening to identify a way to cure the disease by genetic way.
Could you take a moment and describe what a suppressor screen is? What does it mean to introduce a suppressor mutation?
The suppressor screening means we introduce second mutation into the disease model worm and look for better suppressor mutant. Suppressor mutant means the disease model worm shows movement defect. However, the suppressor shows better movement, meaning that the second mutation makes the symptom better. So we expect that if we can identify suppressor mutations, and if we can make inhibitors for these suppressor genes, we think that should be a good drug target to treat CAN.
You've also focused significant research in the recent past looking at how healthy KIF1A and mutant KIF1A operate together. Could you speak a little bit more about what these heterozygote worms can teach us about KIF1A function?
As you know, CAN is caused by dominant mutation. People have 2 copies of KIF1A gene, but if you have a mutation in 1 copy, people suffer from CAN. However, another copy is normal. In this situation, what happens? Our question is, what happens in this heterozygote situation? So we made heterodimers of normal KIF1A and disease-associated KIF1A and measured the motility. We found disease copy inhibits the function of healthy KIF1A, meaning that if you knock down disease copy of KIF1A, that should be a good way to treat CAN.
Are there any other thoughts or messages that you'd like to share with our KIF1A family community?
Yes. We are now looking for a way to treat CAN by genetic screening. We now think one very good target to treat CAN is the auto-inhibition of KIF1A. Generally, KIF1A is inhibited by auto-inhibition. That is like a brake of KIF1A. CAN people suffer from CAN. CAN people have a mutation in KIF1A, and the mutated KIF1A have defect in the motor function. However, even if the motor function is inhibited, if you have mutation in the brake, if you have defect in the brake, the double mutant KIF1A have better function. meaning that if we can make an inhibitor for the break of KIF1A, we can, I think that is a very good target for CAND. We are looking for such a drug. And we think we are now doing a genetic and drug screening using C. elegans to look for a way to treat CAND.